Okay, you guys ready? Great. Thanks for the enthusiasm. I'm going to take that chagrin as enthusiasm. So today we're going to talk about membranes and kind of finish up this. And you'll see that a lot of what we're talking about today, we've actually covered the previous 2 days. So let's start with a patient here. So Edward's a 35-year-old white male. He came to a neurology clinic for evaluation of his long-term neurological complaints, noting that for a lot of years he noticed significant changes in his neurological function, particularly heat intolerance, stumbling gait, and tendency to fall. His eyesight also seemed to change periodically during several years. And 2 months ago, he was working very hard, was under a lot of stress, got sick with the flu, and it got worse. So at that time, he couldn't hold objects in his hands, had significant tremors and severe exhaustion, and several bad falls. So what do we think is going on with Edward? Is there like a general class of things that maybe is going on with him? ALS, yep. So kind of a neurodegenerative type disease. Parkinson's, yep. Tremors, neurological. I think so, yeah. Okay, so we've got ALS and we've got Parkinson's. Both are incredibly good guesses. It's not Parkinson's because Parkinson's doesn't typically affect your eyesight periodically, that I know of anyway. So we can probably rule out Parkinson's, but ALS is probably a pretty good guess. It actually turns out to be multiple sclerosis. So they did a spinal tap, which revealed the presence of this oligoclonal bands. And I'll show you a picture of what that looks like later. And then they did a neurological exam. They did a visual response testing that showed abnormal response with a slow conduction in the optic nerves, and an MRI scan was performed that revealed multifocal white matter disease and a T2 signal in both hemispheres, which indicates multiple sclerosis. Now, one of the first things that people see with multiple sclerosis is often changes in visual acuity. So my brother, when I was in well, actually I think I was married, he was in high school, he was dating a girl whose mom had MS, and she was wheelchair bound, and I think Ashley was probably 16 at the time. And all of a sudden, you know, she actually took care of her mom. She did a lot of the caretaking for her mom, so she saw this disease progression. And all of a sudden, she couldn't see out of her right eye. And she didn't tell anybody, because that's scary. You can't see out of your right eye. So finally, she told my brother that she couldn't see out of her right eye. And she went to the eye doctor, and they saw that there was something wrong with her optic nerve. And so that started her down this path, and she was diagnosed with multiple sclerosis when she was 16. That's a pretty devastating diagnosis, and this is the exact thing that they did. They did a spinal tap, they did an MRI, and it was a pretty easy diagnosis. So here are the oligoclonal bands. What kind of technique are we looking at here, guys? Is it a Western? Is it, what kind of technique? It's what? PCR? It could be PCR, yep. An electrophoresis, yes, you're right, it's an electrophoresis, so we're using electrical charge. It's probably not PCR because what we're looking at is cerebral spinal fluid and plasma, both of which we're going to see a lot of proteins and not very much nucleic acid, and PCR is nucleic acid. So probably we're looking at a Kumasi blue SDS page gel. And so what you see here are these bands, and they're a little bit difficult to see in the cerebral spinal fluid, but we always run our normal control. So we know we shouldn't see bands there, and so we're comparing our normal to our abnormal. So we see these bands. That was the first thing. So multiple sclerosis affects approximately 2.5 million people in the world. There are about 200 new cases diagnosed each week in the US. This is something that at some point you might come in contact with one of your patients. You can get it at any age, but most people are diagnosed between 20 and 40. My brother's girlfriend was diagnosed much earlier because she knew what to look for. The ratio of women to men is 2 to 1. And this is interesting because it's not really known to have this genetic component in terms of we can't find a specific gene, but there is a genetic component in that it runs in families. So if you have a parent or a sibling with MS, you have a 1 to 3% increased chance of developing it. If you have an identical twin with MS, your risk is 30%. So there is a genetic component, and we don't quite understand what that is. Again, women with MS experience relief from symptoms during pregnancy eventually, or a lot of times. But after pregnancy, after they have the baby, there's a high likelihood that they will have a relapse. Because remember, MS is often this relapsing, recurring type thing. And so there's a high risk of a relapse after the baby's born. And this is the cost per patient per year. Now this is interesting because this cost, some of the medications, some of the new medications that they're coming out with for MS are like $50,000 a year for this medication, which kind of brings up this really interesting point of we can make these fantastic medications, but are they going to be inaccessible for a general population? Are they only accessible for like the top 1% of people? Anyway, so this is pretty significant. And then you can see that there's a couple other comorbidity type conditions that could actually increase your risk. So here, about 63% feel numbness or tingling, 40% have vision problems, approximately 50% have walking difficulty. So you can see there's a variety of different symptoms which makes it difficult to actually understand and tell what's going on. So going back to our first discussion, is it ALS, is it MS, is it Parkinson's? A lot of these have very similar type symptoms. So here's our mechanism right here. We're looking at a defect in the myelin sheath, which surrounds the nerves. And you can kind of think this of an insulation for a wire, okay? And that insulation allows the electrical signal to be conducted down that wire and to reach its destination with a lot higher fidelity. And so the signal can be lower and it still stays in one place. So what happens is when you have MS, the myelin gets damaged and it gets scarred, and so it gets kind of degraded, and it doesn't provide this fidelity, so your electrical impulse comes along, it hits this first spot, maybe a little bit goes through, but not all of it, and eventually this is a progressive disease often, and so the progression really gets rid of the myelin sheath so that you can't control any of those muscles. When we look at the structure, we can see that it's a very complex bilayer membrane. You can see how it kind of wraps around that axon. And here's your myelin sheath showing in the neuron. Here's your cell body. You've got this node of Ranvier right here. In the bilayer, you have your lipid part. Then you have a myelin basic protein that kind of gets sandwiched in between the layers. And you have this proteolipid protein that appears to be more of an integrin type related protein because it's directly into the bilayer. And so what we're going to see today is how all these components come together. So we've learned about lipids and we've learned about carbohydrates. How are they going to come together to make these membranes? So there's some common features of membranes that we should know that we just saw in this particular case of a myelin sheath. And remember, the myelin sheath is the membrane in this case. So a couple common features. They have this kind of sheet-like flexibility, and this is approximately their thickness. The main structure is 2 leaflets of lipids, which gives you this bilayer type look to it. And then they can form spontaneously in aqueous solution. Because of that lipid component that's got hydrophobic and hydrophilic, we're going to see how those will spontaneously form a bilayer because they want to bury those hydrophobic residues. And then they're stabilized by non-covalent forces, particularly hydrophobic forces, because again, we're trying to prevent those hydrophobic tails from coming in contact with the aqueous milieu. There are several different types of proteins. A lot of them span the lipid bilayer, so they go right through that lipid bilayer. There's a lot of asymmetry in the lipid bilayer, so some lipids are found preferentially on the inside of that bilayer, so on the inside near the cell, and some are on the outside. Carbohydrate moiety, so if the lipids are modified with carbohydrates, those are always going to be on the outside. Why? Why would carbohydrates always be on the outside? Think about what we talked about yesterday. What do carbohydrates do a lot of the times? So they could keep things from binding, but what else could they do? Recognition, yeah. So you could either keep things from binding or you could promote binding through recognition. But they really kind of mediate these binding events a lot of times. So they're going to be on the outside of the cell. They're electrically polarized. So the inside usually has about a negative millivolt charge, negative 60 millivolt charge, whereas the outside does not. And then they're very fluid structures. So because they have this two-dimensional orientation of lipids, there are certain proteins that kind of move through them in a lateral orientation. And that's a fluid model, and we'll talk a little bit more about that. So again, here's our general membrane structure that we just talked about. We have a couple of different types of proteins here. You've got some cholesterol that sits in here. Here's our globular proteins. We have peripheral proteins that we'll talk about today. We've got glycolipids, so here's our sugar residues. We have different proteins that are spanning the membrane. And a lot of times those are going to be for transport. So we're going to use those to transport molecules into and out of the cell. And those are going to be kind of our gatekeepers and regulators. We have a lot of our carbohydrates over here. We have an alpha helical integral protein. So integral proteins are different than peripheral proteins. And peripheral proteins and surface proteins are often considered the same. And you can see that we've got our hydrophobic tails that are buried in the center of this bilayer. This is what it actually looks like. When you look under an electron microscope, this is what you see. Remember that when we're talking about membranes, we're not just talking about the plasma membrane that surrounds the cell. We're also talking about membranes that are occurring within the cell. So your endoplasmic reticulum, your nuclear envelope, your nuclear membrane, your mitochondria, and here are your secretory granules. And so you have membrane components, and all these membranes have those same kind of characteristics. They have proteins, they have lipids, some of them have carbohydrates on the outside, but proteins and lipids are kind of our characteristic pieces. However, they can have a lot of diversity, depending on whether or not they're inside the cell or outside the cell. So again, you've got those polar head groups and nonpolar tails. The diversification or the difference and what gives you this compositional difference is based on those polar head groups that we talked about, that we looked at a couple days ago. They're also based on changing the fatty acids, so looking at how long is that carbon chain, how many double bonds does that carbon chain have, and seeing how that's going to affect the particular membrane. So the properties of the head groups, because they're what's sitting on this outside here, are going to dictate really what's going on with that membrane and how it's going to interact. And then remember that different organisms have different types of lipid membranes because of those different type of head group compositions. And then if you add the carbohydrates on, because we saw how Some organisms can't add sialic acid, and some can't add fucose. So the carbohydrates are also going to provide some diversity from the organisms. And then finally, different tissues have different membrane lipid head groups as well. So not only do you have differences between organisms, but you also have differences in tissues. So when we look at the composition, there again are just a few basic characteristics. So we have our phospholipids and glycolipids. So here are the principal membrane lipid components that are shown right here. And then we also have our phosphatidylserine, which is a phospholipid, that's shown here. You've got cholesterol, which is the sterol usually when you talk about mammalians, and you'll see how that differs a little bit from organism to organism in one of our charts later on. And then we have our glycolipid here. So here's a look at how some of these things can vary based on where the membrane is found. So we can see that here's a composition of 6 different types of membrane components, where you've got cholesterol, you've got phosphatidylethylamine, you've got your sphingolipids. So if you look at your plasma membrane, which is oftentimes what we think that we're talking about only, is just that plasma membrane that controls what's going in and out of the cell, you can see that there's a pretty even distribution, and you have a lot more cholesterol than any other place. You also can see that you've got pretty equal amounts of your sphingolipids, your phosphatidylcholine, your phosphatidylethylamine, and then you have this minor lipid component that they don't talk too much about. The cardiolipin is completely absent in the Golgi. So you can see that there are differences between these different components and between the different compartments that we have inside a cell.
No one's going to ask you to memorize.
No, please don't memorize composition. Just know that composition differs based on where you're looking at the membrane. So here's the different sterol types that you can see. So this is myelin sheath that we started out looking at. The sterol there is cholesterol, and 19% of that myelin sheath is cholesterol, which tells you that it's probably fairly rigid and is going to act as a very good insulator in general. So when you look at a mouse liver, 25% is cholesterol, but then that's where we depart from cholesterol because we're getting into plants and different single-cell organisms, and in fact, E. coli has no cholesterol or no sterol at all. So as promised, these lipids form aggregates, and they aggregate in such a way as to protect those hydrophobic residues from interacting with an aqueous milieu. So what they form is what we call a micelle. And we talked a little bit about micelles the other day versus nanoemulsions, and they're very, very similar. In fact, I think probably people would use them interchangeably. Micelles, though, often have this outer and then an inner polar head group region. So you have this hydrophobic layer that's coated with basically your hydrophilic layer, and then you have the hydrophilic layer in the center, and they call this a vesicle and this is a micelle, but they're used very interchangeably. So some people will call this a micelle and not even use the term vesicle. So just realize that it's kind of, they play loosey-goosey with the terms there on that. Each individual unit kind of forms this wedge shape because you have a hydration sphere around your polar head group. It forms a wedge shape, and then that also allows for a tighter packing within the center of the particle. So it allows for this really tight packing of these polar head groups to exclude water or the aqueous milieu. So that's essentially how a membrane bilayer forms. It kind of has this self-assembly type process where you have a hydrophobic effect that drives the formation of a bilayer. So here in a membrane bilayer, though, the difference is that the individual unit is in a cylindrical cross-section, which means that the hydration around the head is the same as the side chains. So here's your bilayer here. You've got two leaflets. You've got a complex lipid base. These are very pliable. You've got a variety of different kinds of lipids. You've got cholesterol that's embedded in here, and we've seen a lot of these things before. Keep in mind that these membranes are not necessarily uniform, so you can have different things on the inside and the outside. They're not symmetric, and they have different compositions. So the outer leaflet is usually more positively charged. Why would that be? What do you guys think that is? What did we find out about the inner versus the outer in terms of electrical properties? Approximately negative 60 millivolts. So we've got the outer leaflet is more positively charged. Now, phosphatidylserine is interesting because if it's on the outside, depending on where it is, it's going to do different things for you. So if it's on the outside of a platelet membrane, it's going to be activating. And so you're actually going to see more blood clotting. But on the other cells, it can actually mark the cell for destruction. So it can do different things based on the type of cell that it's on and where that cell is found. And you can see kind of our distribution over here. So again, we can have this membrane asymmetry. Here's our two monolayers that can differ in different compositions. So here we have our cholesterol and our sphingolipids kind of on the outside, and then we have our phosphatidylethylamine and phosphatidylserine on the inside. So we can get this very complex type membrane environment. So there are, in some cases, some people classify it as 3 major functions, some people classify it as 6. So here we can see that we've got receptors, which are going to be over here. We've got our channels, which are shown over here for transport, and we've got our enzymes shown right here. Now there are certain texts that will add in cell-cell recognition, intracellular joining, and attachment of cytoskeleton compounds. So those are, some people consider them base functions, some people consider them secondary functions. Just know that they are possible functions. But these top 3 are going to be our primary ones. So we have receptors for hormones. We have receptors for light. We have receptors for neurotransmitters and pheromones. We have receptors for everything. And we're going to see some of those receptors when we talk about biosignaling later today. We also have channels, and those channels can be considered gates and pumps, and they transport sodium and potassium and basically all sorts of nutrients and neurotransmitters and glucose. And so we've got transporters for everything. Now, a lot of transporters are very, very specific for their ligand. We'll see that later today too. And some of them are not as specific. So they're looking for just general characteristics. This ligand has this general charge. This ligand does this specific thing. And they're not as specific for ligands. And you'll see this when we talk about ABC transporters. And then we have our enzymes. And a lot of the enzymes are going to be involved with lipid biosynthesis. And then you're going to have a lot of them involved with ATP synthesis. Because oftentimes ATP is going to drive our ion channels. So it's going to drive our transport. So we kind of have to couple those things together. Just like we talked about when we looked at how do we lower activation energy, we talked about sometimes we have to couple those 2 reactions together to make one more favorable. And then keep in mind, you'll learn this in a later section, but it's an F0, F1 ATPase, ATP synthase that does this. I don't think you're going to see that on tomorrow's test, but you'll definitely see it later on. So let's start going through some of these different proteins. Peripheral proteins are interesting because they are loosely associated with that lipid bilayer, and they just kind of hang out on the outside, or the inside in some cases. So they're very loosely associated with those polar head groups, and usually that's through ionic interactions. They can be removed by disrupting those by either high salt or a change in the pH. You can lose your peripheral proteins with one of those two functions. So if we change the pH, you can actually change your peripheral proteins. Why does that matter to you guys? What do we talk about in terms of if we change the pH, what happens to medications? Yeah, it changes the way they metabolize. It changes the way you excrete them. If we're changing the way we excrete things and we're changing the way things metabolize, we may actually be affecting our peripheral proteins, right here and right here. And then purified peripheral membrane proteins don't have to be associated with lipids. So it's not like the lipids go with the purified peripheral protein, whereas they may if you purify an integral protein. And we'll talk about that here in a couple minutes. So peripheral proteins, loosely associated, they can come off. They're associated through ionic interactions, and they have a variety of different functions. Now, if we compare that to these integral membrane proteins will span the entire lipid bilayer. A lot of times they have alpha helical domains that are going to sit in that lipid bilayer, and those are called transmembrane domains. Sometimes we'll have beta transmembrane domains, and a lot of times we have alpha transmembrane domains. So these are going to be really tightly associated with the membrane. You really can't purify them away from the membrane lipids. That's very, very difficult. And so there are these hydrophobic stretches, and that's what allows them to go through the membrane. You can remove them if you destroy the lipid membrane. But if you destroy the lipid membrane with a detergent, you risk also destroying the transmembrane domains. So these are really, really difficult to crystallize. There's only been a couple that have been successfully crystallized, and so some of the data on these in the PDB may not be as high of quality as some of the stuff that we've seen. But we can still see a couple of these. So those purified integral membrane proteins, again, you almost always have phospholipids associated with them, because the space that surrounds these alpha helices are going to be filled with those acyl chains. So they're going to be filled up almost, and you just have this really tight interaction. So here we have our amino terminus on the outside, and we have our carboxy terminus coming through the inside, and then we have 7 transmembrane domains. For some reason, 7 transmembrane domains seems to be very common. There are 6 different types of integral proteins, and somebody clever just named them type 1 through 6. So you can see type 1 has your carboxy terminus on the inside and your amino terminus on the outside, and type 2 is exactly opposite. Type 3 has your multiple transmembrane spanning domains here, but they're all connected. Type 4 has this kind of integrin type approach to it, or a ligand that's sticking into the membrane but doesn't go all the way through. Type 5 has multiple transmembrane domains, but they're from different proteins, and they're all going to associate to form a single structure. And then you have type 6 over here, where you've got a transmembrane domain, and there's only one of them typically, and then you're also anchored into the membrane here. So you're anchored on the outside typically. Questions about the different types? You'll want to know those different types and what distinguishes them. So then we can have amino acids that occur in protein clusters in the membrane, and they occur in very specific ways. So if you look at tyrosine and tryptophan, you've got this, they usually cluster at a nonpolar polar interface. So you can see them kind of clustered right around here. Tryptophan is going to be in red, tyrosine is going to be in kind of this orange color. And then charged residues like lysine, arginine, glutamine, and asparagine are found almost exclusively in the aqueous phase. So they don't want to be in that charged region. So that's typically how that happens. Now, we can see a lot of these different kind of segregation of amino acids when we look at our potassium channels, this maltoporin channel that transports maltose through a cell. You can see it in phospholipase, and then in a couple other ones as well. Here's some beta sheets that we can see. So we just looked at our alpha helices. We can also see these beta sheets, which form these really kind of cool beta barrel structures almost. So it's like you're looking down the center of a barrel into the cell. And then we can transport things through there. These ones are all in an E. coli. FEP-A transports iron-containing compounds that are really, really important for bacteria, and these are called siderophores a lot of times. So those iron-containing compounds, it's pretty interesting. One of the ways that we're actually looking at trying to combat some antimicrobial resistant bacteria is by attaching things to siderophores, because it's really hard to get stuff into the center of bacteria, to get a particle or anything else in there. So one of the ways that scientists are looking at doing this right now is by coupling it to their iron compounds, because bacteria have to have iron to grow and proliferate. So if we couple it to something they absolutely have to have, then they're going to take it in themselves. And we can actually stop a lot of antimicrobial resistance. At least that's the theory. Here you have the maltoporin again, where you can see that you've got this really nice kind of beta barrel structure, and that's for transporting maltose. You also have these lipid-linked membrane proteins. So just like we saw with our really tightly kind of integrated proteins, we also have lipid-linked membrane proteins. So this looks more like a type. Which type? So if we look at the types, which type would you guys think this is? Type 4? That would be my guess too, because we don't see it wrapped back around. So we don't see that loop. So my guess is that that's a type 4. So this allows us to target certain proteins. So if we can put these proteins on the outside of some of our drug carriers, we could target them to certain cells, for example. So it allows targeting of proteins, and some of these GPI anchors that we'll talk extensively about are G protein integrins. Anchors are only found on the outer face of the plasma membrane. So this is probably a G protein, which I believe is a type 4. And then here we have a couple of different types on the inside. And you can see that they're kind of linked in here. So now let's go back to our multiple sclerosis study and talk for a minute about, we know we're now affecting the myelin sheath. We know that that's affecting a membrane structure. So how do we treat that? What do we do to treat an effect of a membrane structure? So here's one particular drug, and we can tell it's an antibody because it ends in MAB. So this is a monoclonal antibody. M stands for monoclonal, meaning that it's against one particular type of clone. And it's against the cell adhesion molecule alpha-4 integrin. Now, we haven't talked a whole lot about integrins, but I'll show you integrins in a couple of minutes here. Integrins, as you can imagine, are going to be very integrated in the plasma membrane. So we're going to target this adhesion molecule, alpha-4 integrin. And this can also be used for Crohn's disease. So again, we think that we're going to reduce the inflammatory immune cells because this is an autoimmune disease. So your immune cells are attacking your myelin or attacking your intestine in Crohn's disease, and allow them to pass through cell layers lining the intestine. So first we'll talk about integrins quickly. They have this transmembrane receptor, and these are kind of that bridge for that cell-cell communication and cell-cell adhesion, and they can trigger these signal transduction pathways. So I really like this example of an integrin. And the reason why I like it is because it shows you how drastically integrins can change when they bind something. So this is, you can see your alpha chain and your beta chain here, and you have sharpen down here, which is just a protein, and that doesn't really matter. But when talon comes in and binds, you absolutely completely change the structure of that integrin. That's a wildly different structure, which I think is pretty cool. So this is the inactive form, and this is the active form. So if we are going to target this, we need to figure out if we're going to target it as an active form or an inactive form. Well, we don't really want it adhering to different things because we're going to target it on the immune cells. So here's our proposed mechanism. Down here, we're targeting this alpha-4 integrin, which is going to prevent it from interacting with immune cells, and it's going to pass the blood-brain barrier. So that's how integrins work. So now let's talk about these cell membranes are very asymmetrical. We've already talked about electrical asymmetry. So how else might they be asymmetric? So again, the lipids on the outer and inner leaflet are going to have different lipid compositions. We saw that. You're going to have different proteins. The proteins are not going to be the same. Here we're looking at a channel protein, carrier protein, and a pump, all embedded in a single membrane. We're going to have individual peripheral membrane proteins only associated with one side of the membrane. So we could have a peripheral protein sitting right here, or maybe we have it sitting over here, or maybe all the peripheral proteins are on one side, or vice versa. We also can have different domains on different sides, and we can do specific lipid modifications only on a specific leaflet. So what modifications have we already seen that are only on the outside? Remember we talked about them for cell recognition. Yep, your carbohydrates. So those sugar residues are only on the outside. So that's what they're talking about there. Our modifications are associated with one side or the other. And then that goes to those carbohydrates only being on the outside of cells. And this is just showing you the electrical chemical potential gradient, how it's high versus low. Now this gradient is really what drives what can come into and go out of your membranes. This is going to become incredibly important. If you can't maintain an electrical chemical gradient across your cell membrane, your cell won't survive. So again, these cell membranes are not permeable to large polar solutes, but they're permeable to small things. So this diagram right here is showing you these small hydrophobic molecules like oxygen, CO2, nitrogen, and benzene will just go right in. Small uncharged polar molecules also can go through, so that's going to be your water and your glycerol and ethanol. But your larger uncharged, like your amino acids, glucose, and nucleotides, are going to be repelled. So this is considered to be a semi-permeable barrier because it lets certain things in and it repels other things, and your ions are also going to be repelled. Now this chart down here shows you that we have increasing permeability. Here's our permeability coefficients on the bottom. And so things that are on this side of the chart are going to be able to go through, and things that are over here are not going to go through nearly as quickly, if at all, without a transporter. So that's how you're looking at these different charts here. And a lot of this permeability and a lot of the fluidity of membranes is going to be based on the sterol that's going to be embedded in that membrane. Now, we saw that the sterol can change based on the organism, but we're going to focus more on cholesterol. So if we have cholesterol, remember you've got cholesterol in animals, phytosterol in plants, and eosterol in fungi. And so when we have cholesterol, it depends on whether or not it's saturated or unsaturated. That changes the temperature. Remember, we talked about yesterday if it's going to be a fat or an oil. So the structure of the cholesterol is going to change the fluidity of your membrane. So again, depending on how much cholesterol, you're either going to have this kind of gel or a fluid phase. So in a gel phase, we're talking about, now we saw TM before when we talked about PCR, right? We saw it was like an annealing temperature. This is your glass transition temperature in this case. So this is the temperature at which things melt. So in a gel phase, the individual molecules don't really move around. You can see they're pretty steady. But when we embed our sterol in there, a lot of times we get this liquid phase, and it's very ordered. And this is the basis for this fluid model that we're going to talk about, the way things move through membranes. But if you are heating it to go from a gel to you have to go heating to go from a gel to a liquid. Or you can have a liquid ordered over here, and starting from a liquid disordered. And this is when your temperature is greater than this transition temperature. So remember, under physiological conditions, you're going to be more liquid than gel. Why would you not want to be more gel? What's going to happen if your cell membranes are more rigid? UA? Yeah, so it could change your permeability, and you might not be able to get things in that need to get in. You also change the way things move through your membranes, and so both can have a significant impact. So that's why organisms a lot of times will adjust the composition of their membranes. They need it to be more fluid for a variety of reasons. So that fluidity, again, is determined by cholesterol, which is determined by the fatty acid. And what do we know about fatty acids? What determines if they're going to be a wax or a liquid, or what determines that? There's two parameters. Length. Yep, the bonds, saturation. So how many double bonds, how many kinks do they have? So that fluid membranes require shorter, more unsaturated fatty acids. And then at higher temperatures, cells need more fatty acids in order to maintain that integrity, so they want it to be a little more stiff. At lower temperatures, cells need more unsaturated fatty acids to maintain fluidity. So it kind of depends on temperature, which is why I think this would be really interesting, because have you ever wondered how people survive in like really significantly hot temperatures like Phoenix? I mean, there's a reason I don't live in Phoenix. It's really hot there. So, but some people adapt to that very well, and they adapt very, very quickly. And so I wonder what their cholesterol looks like. Does it change their diet? Does it change how their cells and the rigidity of their membranes? So I keep referring to this fluid model. It's called the fluid mosaic model. It was first proposed in 1972 by Singer and Nicholson. And essentially it says that these lipids form this really viscous two-dimensional solvent. So think of your lipid bilayer as a solvent, not as a solid. And that you've got proteins that are inserted into it at a certain depth. And some of them are going to be inserted at a greater depth than other ones, but they're all going to be inserted, and because this is now a liquid, that they can move back and forth. So integral proteins firmly associated with the membrane, we know that that is the case, and that has to be part of our fluid mosaic model. When you develop these models, you can't just say, oh, well, it's not going to be associated because that's what makes my model easier. You have to go with what's physiologically the norm. And then peripheral proteins are weakly associated and easily removed. Some are non-covalently attached, and some are linked to membrane lipids. So some are going to be linked, but they can be removed. So here's our fluid mosaic model of membranes. Essentially, again, you have this kind of lateral diffusion ability within your lipid bilayer. You've got your peripheral proteins that are covalently linked to lipids in some cases, and then in some cases they're just ionically associated with the polar head groups. You've got your integral proteins, you've got carbohydrates sitting on the outside, and then you have other integral proteins. You've got GPI anchored proteins into the membrane, and so forth. So within this model, there's a dynamic component to it that says that individual lipids can undergo fast lateral diffusion within their leaflet, but they very rarely will flip over into the other leaflet. So they move very fast back and forth. But spontaneous flips are rare because in order for it to spontaneously flip, this polar head group has to go through this unpolar or hydrophobic tail region. And so flipping it is not very easy. In fact, it takes about half a day to do that. So one of the things that the model proposes are these rafts. So think about it, you're in this lipid ocean and now you're on a raft. So these lipids are distributed in not a random order, but it's very uniform. And these lipid rafts are clusters of glycosphingolipids that have very long tails. So these rafts, you can see how our membrane kind of bulges out right here. It's because these tails are longer. These are more ordered than the rest of your membrane, and they contain these doubly or triply acetylated proteins. So they contain different proteins. You've got your cholesterol in here, and this allows segregation of proteins in the membrane. So I can make a lipid raft, and then I can segregate my GPI protein in that lipid raft. And then that whole thing can move laterally through your membrane. So you can move your lipid raft. So because we have these lipid rafts, this is what we've now talked about, a bilayer. I've shown you how lipids can spontaneously form kind of these spherical structures, but not necessarily a bilayer. A bilayer is a little more difficult. So Catalin specifically will come along and it makes the membrane have this invagination right here. And the way that it works is by acting specifically on the lipid raft. So by acting specifically on that lipid raft, you can actually get this invagination. Now, the invagination can give you additional surface area in some cases, but it can also make the entire cell concave. So here's our Catalin dimer over here. It's got 6 fatty acyl and 8 cholesterol moieties that sit right here. And they interact with your lipid raft to make you have this kind of invagination. This is also the basis for how membranes fuse. Now we saw yesterday, I think, was it yesterday that we talked about HIV? I think so. Anyway, we saw that this happens a lot with viruses, right? Viruses have to be able to fuse to a membrane in order to transmit their genetic information. Well, this happens with other things as well. We need things to be able to fuse to our membranes in certain cases. So we can see that membranes can fuse with each other without losing continuity. So here you have separation of 2 plasma membranes at cell division. They can fuse. We see this with fertilization when the sperm and egg meet. We can see fusions as spontaneous or protein mediated. In some cases, there's a receptor that we're going through. We can also see them with entry of influenza or basically any virus. We can see it with release of neurotransmitters. And then another example that might be a little bit more relevant to you guys is these liposome drug deliveries. So when we encapsulate a drug within a liposome, we're relying on the fact that that liposome has kind of this bilayer type structure with the drug encapsulated into it. And because of that bilayer structure, it can then fuse with the normal plasma membrane and release its drug into the cell that we need it to release into. We can also transport things across the membrane. There are a couple different ways that we can do this. We can have a simple diffusion. So this is only going to work for nonpolar compounds, typically, and only going to work if you're going down a concentration gradient. So you can't simply diffuse and go up a concentration gradient. You can have facilitated diffusion. Again, you're going down a concentration gradient. You can have primary active transport. So here we can go against a gradient, and these are going to be driven by the use of energy. So when you think about anything active, you've got to have energy driving it. So this is driven by ATP. You can have a secondary active transport. So you're going to couple the diffusion of an ion with some other substrate. You can have an ion channel. Again, you're going to move down the electrochemical gradient, and this can be gated or ungated. And then you can have just ionophore, which again is going to mediate ion exchange. And this has to move down an electrical chemical gradient. So you're going to want to know these 6 different kinds. So if we have polar solutes, so S in the other, in this figure is solutes. So if we have polar solutes, we really need to provide an alternative pathway for them to cross the membrane, because they don't just cross it on their own. So the alternative pathway that you can see, and here's our free energy diagram again. So this is without a transporter, and you can see that our energy of activation is very, very large. And with a transporter, we decrease our energy of activation. And so here's our transporter here. So this may happen occasionally, but there's a huge barrier to making that happen. So typically we have a transporter that's going to work in conjunction with that. There are 3 classes of transporter systems. There's a uniporter, a symporter, and an antiporter. Now, sim and antiporters are considered co-transporters or co-transport because you're moving 2 molecules at one time. In sim, they're moving in both the same direction. In antiports, they're moving in opposite directions. And then uniports are just moving a single direction, and they're just moving 1 solute at a time. So that's the difference between these 3 different classifications. So if we look at glucose transport, here's our GLUT1 transporter in the membrane. You can see here's the amino acids that give you that. Here's the amino acids that give you that, and then here's another look at that particular molecule, and you've got your glucose right in the center. You can see your transmembrane domains and your loops going through here. We can see that we have both our amino terminus and our carboxy terminus on the same region. We have a lot of hydrophobic regions here, so these are going to be our alpha helices transmembrane domains typically. And then you've got polar and charged groups that are going to be on the inside and some on the outside. So here's our model for glucose transport. Typically you have a T1 and a T2 conformation. You've got D-glucose that is going to come and sit to this T1 site. That T1 site is going to allow that in and clamp down and convert to a T2. So it's going to change its conformation. As a T2, this is going to move out and transport into the inside. Sometimes this requires secondary transport. So secondary transport is where it's going to couple with the transport of typically sodium. Sometimes you'll have transport coupled to potassium, but typically it's going to be sodium. So here you can see our sodium-potassium pump. We have 2 potassium ions coming into the cytoplasm for every 3 sodium ions going out. The sodium ions then come over into this glucose transporter and transport with glucose at the same time. Glucose will not transport if you don't have the sodium. So that's why this is a secondary transport. That sodium gradient, because you're pumping 2 or 3 out and then 1 in, that gradient is what's driving the transport of glucose. Bicarbonate is the next really big one. Bicarbonate we know is essential for a lot of our cellular functions. This is what allows us to maintain that electrochemical potential. So here's our carbon dioxide that's produced that's going to enter through the erythrocyte. Here's carbon carbon dioxide. If I could speak today, that'd be better. Here we're in respiring tissues. We have carbonic anhydrase that acts on our carbon dioxide and water in order to change over to bicarbonate. Now we have bicarbonate that's going to go through the bicarbonate exchange protein right here, and it's going to exchange with chlorine. And then that's going to go back the other way. So the bicarbonate or the chloride bicarbonate exchange protein is incredibly important in maintaining the pH and in helping move carbon dioxide in the lungs. So those are a couple examples of co-transporters that we talked about. There are two different types of active transport. We have our primary active transport and our secondary active transport, and our glucose sodium is going to be secondary active transport because that sodium is going to be moving down its electrochemical gradient, which is going to be helping to actively transport glucose. Here's our primary active transport, where essentially you're just using a kinase and you're basically stripping off one of your phosphate molecules to use to create energy to transport your solute. Over here, this requires two different transporters, so think about secondary is going to require two. You've got two transporters that are going to be necessary to move these solvents or these solutes around. You've got solute 2 coming in, and that's going to help solute 1 based on its electrochemical gradient, because there's a lot more outside the cell than inside, and it's going to move through it. So this is going to be a symporter because they're moving in the same direction, and this is a uniporter. Why are you laughing? This one. It's cool. Okay, I'm going to let you describe it because I'm going to have a coughing fit here any minute. You got me sick, you guys.
Your sister got me sick, and Jess got me sick, so I blame you. So there, it's all your family. Just returning the favor. All right, and that's why I sound so bad today. So this protein is the last protein in the electron transport chain, which is what we use to make ATP, which is the energy of the cell. So just put it into context, okay? And so this is the workhorse for making ATP. And it takes a proton gradient, okay? So you're going to pump protons, okay? And that's going to cause the manufacturing of ATP. It works both ways. So it can go either way, and it can either pump protons out, or it can take protons in, and it can make ATP, or it can use ATP. So this is just basically a reverse, and then we're basically using, in a mitochondria matrix, we're going from a higher concentration of hydrogens to a lower concentration of hydrogens. So you're just pumping it across, and you're using that potential that energy being used, and this thing is a molecular motor, okay? And this is one of the few lectures I remember from biochemistry when I was an undergraduate, right? So this is the F1 part of the motor, and this is the FO part. So some people call it F naught, I call it FO, she calls it F naught. I've heard it both ways. FO, F1 ATPase. You guys didn't laugh at that. Okay, fine. We'll keep it an inside joke then. So FO, F1, AT, this is the FO part. This is you can see that there's different subunits in this. There's a stator here, and then this is a motor. And this actually, it looks like an orange. So these are different proteins that are fit together like an orange peel. If you could peel apart this, it would look exactly like if you were peeling an orange and breaking it apart. And there's 6 different subunits in there, 3 alpha and 3 beta. And then there's a stator in the middle of there. And this thing spins around. It's a molecular motor, and it spins around like you could like you would imagine an electric motor spinning around, and that's what actually that that force is what actually generates the chemical reaction that generates ATP. So this proton gradient here helps generate all the energy in your cell, and this is a motor, and is basically a molecular motor at a fundamental level, which I think is incredibly fascinating, although some people may not.
The cool thing to me is that they've been able to take molecular tweezers and actually tease this apart and watch it flip something around. So they've been able to actually see this motor work, which I always think is really very cool when you can actually see a molecule working. So let's stop here quickly and take a quick break, and then we'll come back and finish this out. Apparently that first part wasn't important. Nobody needs to listen to that again. So like I was telling you, they have, these are really difficult to try to crystallize and try to see all these different intricacies. But they've been able to crystallize about 5 of these guys. That we're showing 4 of them up here. Here's our staph multi-drug exporter. So staph does this great thing where in order to become resistant to different antibiotics, they increase the genetic regulation of an exporter. And so this allows things, and this is one of those ABC transporters. So your ATP is going to come and bind. You can kind of see it binding here. And then that forces drugs out, and it can push them out. You have an importer right here and a fungicide. Here's an E. coli, and here's influenza. And so these are just a couple of different examples of the crystal structures that we'll actually see. So gated ion channels cannot be underestimated in their importance. We have a ton of gated ion channels. These really are kind of the key regulators of how we move ions across cell membranes. So they're going to respond to changes in the membrane potential and to ligand binding at very specific receptor sites. And these have really, really important roles in the nervous system specifically. They also have important roles in the heart. So nervous system and heart are kind of where you're going to see a lot of these. And one of the very important ones is this nicotinic acetylcholine receptor that is going to get hit by nicotine and acetylcholine. Here's a couple different examples of these ion channels. So again, you've got outside, inside, you've got these transmembrane domains. Some of these are really large and have a lot of different transmembrane domains. This one is a potassium one, and this potassium one is going to come in, and you've got this double helix dipole that's going to stabilize it and allow it to move through. And the interesting part is that it allows it with hydrating water molecules, which you don't often see. And the reason you don't see that is because this is very hydrophobic. So it doesn't want anything that has that hydration bubble to actually move through. So most of the time with these transporters, with a lot of the transporters, the first thing you see is a dehydration event and the energy that it takes to remove that solvation shell in order to move things through. So this one is unique because it forms kind of this cage, and it protects the inner membrane, it protects the lipid bilayer so that this moves through with this hydrating water molecule. So again, the inside of the cell typically negatively charged compared to the outside. Membrane potential is largely due to the sodium-potassium ATPase transporter. So for every 2 potassium that are coming in, you're moving 3 sodium out. This is a big part of glucose transport because it hits that GLUT1 transporter that we talked about. That's going to allow sodium to come in because of this imbalance in the chemical gradient to move glucose in as well. So this allows the flow of ionic species across the membrane and allows that gradient to be maintained. So sodium potassium is going to be a really big gradient maker. And that takes us to biosignaling. Now, biosignaling, we're actually going to go through this because we spent too long talking about other stuff. So this is, this gentleman ends up having congestive heart failure. So congestive heart failure is when the heart can't pump enough blood, and the blood moves at a very slow rate. So you end up getting congestive heart failure. Here are your symptoms up here: nausea, fatigue, dizziness, weakness, loss of appetite, swollen abdomen, need to urinate at night. These are usually the ones you're going to see is a lot of swelling and edema because you have so much extra water. So sometimes you will hear congestive heart failure is water on the heart because you really do have just an extra amount of volume there. So the causes are shown up here. Some of these are damage from toxins or illnesses. Some of them are that angiotensin type receptor issues that Ben was talking about. And then here are our treatments here. Most of the time you're using these medications and you're using diuretics. So you're trying to get rid of the water. So a lot of these people on diuretics will end up having to urinate all the time. And that's really, you have to adjust those diuretics and really pay attention to that. And then because coffee is also a diuretic, sometimes that can also cause an additional effect that you need to be aware of. So again, to diagnose this gentleman, they did a physical exam. The neck veins, irregular heartbeat, fluid around the lungs, swelling in the liver and legs. We can also do other tests in terms of looking at the health of the heart. So at 40 years of age, lifetime risk of developing heart failure is 1 in 5. This is a huge problem, you guys. 20 million people worldwide are affected, with 6 million of them in the U.S. It's a progressive condition where the heart fails to pump enough blood. There are certain activities and lifestyle factors that will increase your risk. Here's your symptoms again over here, and here are some of our treatment options down here. So some of those treatment options fall within these categories. You've got your beta blockers, which are going to reduce blood pressure and slow the heart rate. And in some cases can reverse heart damage. Beta blockers are going to interfere with certain lateral flow devices and certain tests that you might end up doing. So you got to be aware of beta blockers. ACE inhibitors, again, these are angiotensin converting enzyme. So you're inhibiting that. These will widen the blood vessel to lower the blood pressure and reduce strain on the heart. You have angiotensin II receptor blocker, which will enhance the heart function. The inotropes, these again are for very severe cases, and these regulate blood pressure. And then your diuretics are kind of one of those first line defenses to reduce the excess fluid. You can take a look at this chart, and you can see that from this chart, we've got 47% of these are enzymes, 30% are GPCRs, 7% are ion channels, 4% are transporters, and then you've got a couple of other things as well. 1% are going to target integrins. So the cells are going to receive signals from the environment, and now you have to transport them into the cell to have some sort of action for you. So again, the cells are going to receive it, and they're going to do either antigens, hormones, neurotransmitters, light, touch, pheromones. These are kind of the signal molecules, and you need it to change the cell in some way. Change the composition, change the function, change the enzyme usage, or cause it to grow, cause it to develop. It's got to change something. So there are 3 general stages. There's a reception, so the signal is going to interact with the receptor. There's transduction, so we're actually going to amplify the signal. And then there's a response. And oftentimes, again, this is an enzymatic type response. There are 5 important features of this process. So the first one is that you have a lot of specificity a lot of times in these receptors. You have an amplification event where you've got enzyme 1 here, and I'll show you how that amplification event happens in just a minute. You've got a modularity here where you've got proteins that have to have a lot of different pieces in order to actually see the response, because those different pieces allow that amplification to occur. You have a desensitization or an adaptation to the receptor, and then you have an integration where you've got two different signals coming in at the same time. The typical ligands are going to be these small ions, organic ligands, organic molecules. Polysaccharides are going to be really important, particularly heparin. Peptides, we'll look at insulin, and then there's a whole host of proteins that are going to act as these signaling molecules and hit a receptor. That receptor induces a signal transduction cascade. So here's our specificity. So you've got this signal molecule that's going to fit inside here. So oftentimes it's kind of this double-edged sword. We have a lot of specificity, but then we have certain transporters that don't show a lot of specificity. So specificity is kind of this weird double-edged sword. But because of that specificity, these make great targets for designing competitive inhibitors, because we can get something that comes in and blocks the site and doesn't allow the signal molecule to come in here. So these are great targets. Here's an example of a specificity. Here's your protein partner. It's got an interacting tyrosine right here, and so it's held firmly in place by multiple non-covalent interactions, and that's what provides the specificity. So you can have a protein-protein interaction that provides specificity, or you can almost have like a binding active site, like you might see for an enzyme. Here's an example of your signal amplification pathway. So this is for epinephrine. The epinephrine comes and hits its G protein linked receptor, or GPCR, and one molecule of epinephrine is starting this cascade. Then the inactive G protein activates itself. Then you get an active adenylate cyclase, and now you have between 2 to the 10th to 2 to the 4th molecules of that. By the time you get down to your active phosphorylase kinase, you're up to 10,000 molecules, starting from one molecule of epinephrine. So you can see this massive increase in the signal. So by the time we get down to the response, it's going to release a massive amount of glucose 1-phosphate, in fact 10 to the 8th molecules of it, starting from one molecule of epinephrine. So these signal transduction cascades can be really, really powerful. When we talk about its modular nature, here we can look at a couple of the different types of modules that we have, and you can mix and match, and you can end up seeing a lot of different types of signal transduction based on this. So this will give us our secondary messengers. Here's our RAS signaling, which becomes very important. Here's a tyrosine phosphate, tyrosine kinase. We hit these tyrosine kinases all the time with drugs in our tyrosine kinase inhibitors that I'll show you in a couple minutes. So one of the things that we can do is an inactivation of G protein signaling, because we've got to be able to turn the signal off. We don't want the signal to always be on. So here's one example of how a G protein can self-inactivate. So you've got your GPCR right here. You've got your GDP that's going to hit and generate GTP, which hits your adenylate cyclase. Now this is where you would first see, if we go back to our pathway here, here's our adenylate cyclase. That is one place where you could actually inactivate the pathway. So one of the things that ends up happening is you get this feedback loop. So again, you guys can read through this, but you end up with GTP bound to this GS alpha, which gets hydrolyzed. So it's this alpha subunit that gets hydrolyzed by a GTPase in order to come in and rebind and essentially start this all over again or stop the signal transduction cascade. So we do have some problems with this whole system, though, and nonspecificity is one of our main problems. So we can get these nonspecific binding events that I've been alluding to kind of throughout here, and some of those are going to be based on hydrophobicity, and some of them are going to be based on charge. And so you see some kind of wiggle room in the receptors. So here's our problem. We got hydrophobic ligands that are non-specifically soaked into the membrane. So we can actually measure the total binding, and we can measure non-specific binding in the absence of the receptor. That will allow us to know how specific that particular receptor is. So by looking at non-specific binding and total binding, we can determine how much of that is specific binding. So this is what that looks like. You know that how much ligand you add. You know how the concentration of your receptor-ligand complex. So this is free ligand. This is bound ligand. You'll see that this is very similar to when we talked about our total enzyme concentration when we looked at our Michaelis-Menten kinetics last week. This is the same thing. It's the total amount that's added. And so we know how much is bound. And then we can determine the amount of specific binding. And that, by subtracting those, we get our non-specific binding. And that will give us a constant. So that gives us our bound ligand divided by our free ligand, gives us a binding constant. And that's based on the number of receptors, the number of sites, and if there's cooperativity. Remember, we talked about that with our heme molecule, with our hemoglobin, where if you bind one oxygen, you're more likely to bind the second one. That's cooperativity that we're talking about. So that gives you your binding curve here, and that slope is 1 over the dissociation constant. So this goes back to our kinetics that we talked about last week, and we can actually determine dissociation constant. You won't be asked to actually calculate dissociation constant on the test, not that I know of anyway. So really the whole point of this is just to know the concept, that if you know the total amount and you know the bound amount, that the bound amount plus the free amount gives you that total amount. So we have the signaling through the membrane. We can do G protein coupled receptors, receptor tyrosine kinases, guanylene cyclases, gated ion channels, adhesion receptors. So there's 5 main kinds. Know these different kinds and kind of be able to distinguish between them. And this is just showing you briefly how they all work. Here's our ion-gated ligand channel, where the channel actually changes in conformation. Here's our adhesion molecules. These are going to be our integrins. You can see that because of this drastic change in the way that they look when they're unbound versus bound. Here's our kinases that set off on a kinase cascade. So these are going to be a phosphorylation, dephosphorylation, which is going to activate or deactivate. So remember, that goes back to those enzymatic, those 6 classes of enzymes that we had. Those are our kinases. Kinases we hit all the time with drugs because it's a pretty easy target to hit a tyrosine kinase. And then you also have these nuclear receptors down here, which we're not going to really talk about too much. Just know that they are nuclear specific receptors too. So G protein coupled receptors, we'll go through this pretty quickly because we've hit a lot of these before. But these are going to be, they've got these alpha helical integral membrane components. They're heterotrimeric, so you have an alpha, beta, gamma complex of 3 of them, and these are going to mediate signal transduction. So GPCRs are going to be one of the major drug targets that you have. Here's a couple of different examples of those. So you have your adrenergic receptor here. You've got your ligand that's sitting right in here. These are going to be very large. This is where your morphine receptors are. So your opioid receptors are going to be G-coupled protein receptors. So here's our mu opioid receptor that sits kind of right in here. You can see it's very small. And you have these transmembrane helices that are sitting right in here. So this is how you're getting morphine in. Here's your histamine receptor. And then this last one over here shows that when you put and you stack 5 receptors on each other, that's what this looks like. Now the reason why we would do that is to show you how much they don't change. So they stay very similar. So they're transporting different things, but they still have a very similar structure to them. So you see transmembrane domains, and then you can see that they overlap pretty nicely. So this is essentially how they're going to look. You've got 7 transmembrane domains. You end up, they respond to light, calcium, small molecules, proteins. You've got an effector or an enzyme channel, and here's your G protein coupled receptor that has the link right in the membrane. This is another look at that structure, just to see it a little bit differently. So epinephrine, we've already looked at. It's one of these big hormone kind of signals that hits our GPCRs. It's going to give you a very strong mobilization of energy and induces cells to break down glycogen. It also induces lipid hydrolysis, and it increases your heart rate. So epinephrine is going to be a pretty big one. This is what it looks like. Here's the molecule up here. Here's your dissociation constant over here. Here's your signaling. Epinephrine comes in, it binds to its receptor here, and then the hormone, the complex causes GDP to bind. So here's your G protein. So GTP is coming over here, binds to adenylene cyclase, which we saw. That turns into a lot of molecules of active pKa and gives you a phosphorylation of cellular proteins. And then we also have the CAMP coming over here. Now, CAMP is considered a secondary messenger in this case, and it's going to get degraded so that it can't go back and restart the cascade. And just, again, another look at how much of a response we can get from a single molecule. So a lot of GPCRs use CAMP or cyclic AMP. To both activate and inhibit. So the human genome encodes about 1,000 GPCRs. They have a wide variety of different molecules that they're sensing. There are also hundreds of different ones that are overlapping. So they're responsible for very similar processes, which should tell us how important GPCRs are to cellular function. Because typically we aren't going to spend a lot of energy making overlapping items if it's not absolutely critical. So here's CAMP. Again, PK is localized, and then you've got a variety of different anchors that come with different cell types and allow you to see what's going to happen to your downstream CAMP. So here's our adenylene cyclase coming here. ATP comes in, CAMP comes off. It comes over here and hits the pKa receptor, which is this duplex. And you've got your secondary messenger, PIP3, in here as well, which will become very important next week. Here's IP3 and calcium. Know that they're also very important for secondary messengers. Here's a couple of different types of sensory perceptions that are really important for being mediated by GPCRs. Vasopressin is a really cool one. Vasopressin helps with how your heartbeat is regulated. It also, you'll notice that it changes with urination at night. So people who have to get up and urinate a lot at night, sometimes their vasopressin is off, which I think is kind of interesting that it hits both of those things. So that one's pretty interesting. So here's our enzyme-linked tyrosine kinases. Here's our ligand binding domain that you can see right here. Here's our tyrosine kinase domain. So again, it just means that we're going to phosphorylate at a tyrosine. Here are all the different types of inhibitors and different things that we have tried in order to hit tyrosine kinases and inhibit their action. By inhibiting their action, we inhibit the on switch a lot of times because they can't go and phosphorylate. And so here are all the different types of diseases that we've tried to hit with tyrosine kinase inhibitors. And here are your different targets. So here's your VEGFR. These are just a variety of different types of proteins. HER2 is one that you see mutated a lot in breast cancer and sometimes in ovarian cancer. So cancers, a lot of our cancer medications, we try to inhibit tyrosine kinases. Here's our GTP or GCMP, or CGMP, sorry. This is going to be activated by protein kinases. So if we're going to inhibit a kinase, you're going to inhibit your cyclic GMP, which again is this important secondary messenger. So what we're looking at right here is this ANF receptor. You've got GTP that gives off CGMP, and then that acts as a secondary messenger, just like our CAMP. So anytime you see kind of that CAMP, CGMP, those are going to be secondary messengers in general. Then we've got our insulin signaling cascade, which Ben touched on and we've touched on before. So here's our GLUT4 over here. This is our signal transduction cascade. This is how insulin works. Insulin comes and hits this receptor right here. And remember, this is a kinase. And so you have this phosphorylation event that starts a specific, you've got your secondary messengers here, PIP2 and PIP3. And so you end up going through and having these secondary messengers that will ultimately lead to glucose release from GLUT4. Then remember that integrins mediate cell adhesion. RGD peptides, you'll see RGD all the time. RGD is the single amino acid code for the peptide that is often used by integrins to bind other cells. So that's going to be your RGD. Collagen, fibrinogen, fibronectin, and a lot of other proteins bind RGD. And that's going to trigger cytoskeleton rearrangement. It triggers blood clot formation. It triggers all sorts of different interactions between cells. This is how that looks. You've got your integrin binding domains. Remember, we have our alpha and beta domains here. And when they're closed or inactive, you see them as kind of bent over here, so they're not actually interacting with collagen in this case. But the collagen in the extracellular matrix triggers that activation. So here's our inactive, and then they change in conformation with the binding of talin and collagen. And that allows this sensing of what's going on in our extracellular matrix. And we already looked at that. We've got our gated ion channels. These are going to respond to changes in membrane potential, and they're very important in a lot of different cellular receptors. Here's how we use ion channels in nerve signaling. Calcium and sodium are going to be really important in those nerve signaling, the way that we're taking things up and the way that it's transporting across the membrane. And here you can see a change in action potential. This is what's going to transmit that nerve impulse between those different cells. So you actually change the way ions are crossing, and you change that through these sensing vesicles. And here you have your postsynaptic neuron over here. Acetylcholine is going to be our receptor ion channel, and then you are releasing sodium and calcium in a lot of cases coming over here, and you have a depolarization event that's propagating this direction. This is what that channel actually looks like when it gets modeled. Here's our extracellular funnel, our selectivity filter, and our central cavity, and our activation gate. We can have voltage-gated sodium channels. This is just another look at that. Here's our inactivation gate with our tether, and then our activation gate is up here, and we've got all of our transmembrane domains through here, which allow us to sense. Those hormones are going to directly hit that, that particular type of regulation. So you have your hormone here, where you can read through, your hormone is going to come in here. It's going to specifically hit. Now, why does the hormone come through, guys? Remember, we talked about this yesterday, how the hormone can penetrate because it's lipid soluble. So your hormone is going to almost act through passive diffusion and come into your cell to cause a change in the DNA. A bunch of different signaling molecules that you should at least be aware of. I think the rest of these slides, this will be the last slide we go through. We already talked about acetylcholine the other day, so this will be the last slide we go through. So this is a voltage-gated ion channel. And as we saw at the beginning of the lecture where we talked about MS, and then we saw in the middle of the lecture where we were hitting that particular receptor with the drug, here we're hitting a voltage-gated ion channel. And this is a potassium channel where we can see that we've got CNS depression over here and hyperexcitability over here. And so we can actually intervene and have a therapeutic intervention based on either inhibiting or activating this potassium channel. So we can hit this particular ion channel, this voltage-gated ion channel, with a therapeutic, or we can try to, in order to regulate this neuron impulse. And that's one of the newer things that they're trying with MS, is to actually regulate this impulse by targeting those ion-gated voltage channels. Okay. Any quick questions? Any long questions? All right, I should be in my office most of tomorrow. If you guys have questions, please come and see me, or shoot me an email, and I'll do my best to answer through email tonight if you need help.